All right, so hi everyone. As Karen just mentioned, my name is Emily, and today I'm excited to share with you guys some of the work that we've been doing here at UWA using rTMS. And so typically, rTMS is delivered to the cortical region of interest at superthreshold intensities, so intensities capable of inducing action potential firing, MEPs if you're stimulating the motor cortex, things like that. But it's also thought that the cortical regions surrounding that target area also receive rTMS, although at much lower subthreshold intensities. And so that's what some of the research here in our lab is currently focusing on. What are the effects of subthreshold rTMS on the brain? And while a lot of studies focus on characterizing rTMS-induced changes in synaptic or synaptic-like plasticity, research from our lab has shown that subthreshold rTMS can actually alter neuronal excitability via changes in action potential thresholds, so the threshold that's required to induce an action potential. And so those findings kind of got us thinking, well, what other neuronal regions is rTMS targeting? Or more specifically, is it targeting the part of the neuron responsible for generating action potentials? And so the part of the neuron responsible for generating action potentials is called the axon initial segment, or the AIS for short. And so this is the AIS here. It sits just proximal to the soma, right after the axon hillock, and it's packed with really high densities of voltage-gated sodium channels, so densities much greater than that found at the soma or at the dendrites. And so it's these really high densities of sodium channels that make the AIS the site of action potential generation. And as what's really interesting about the AIS, as Jamie just mentioned in the previous talk, is that it's plastic, and so it will actually modify its structure as a way of altering a neuron's ability to generate an action potential, which we refer to as neuronal excitability. And so it does this in response to chronic changes in neural or neuronal activity. And so during periods of low neuronal activity, the AIS will move closer to the soma, and or it will increase its length as a way of increasing neuronal excitability and increasing action potential firing. And then during periods of high neuronal activity, it will do the opposite. So it will shift further away from the soma, and or it will get really short. It will reduce its length as a way of reducing neuronal excitability and reducing action potential firing. And so as I mentioned before, this AIS plasticity, it occurs in response to chronic changes in neuronal activity. And so knowing that, one, rTMS induces changes in neuronal activity, and two, that subthreshold rTMS has led to changes in action potential threshold, we thought that rTMS could very well be targeting the AIS and inducing AIS plasticity. And so I tested this theory using a high throughput in vitro system of mouse cortical neuronal cultures. And so after isolating these neurons, I would then plate them onto little glass cover slips and incubate them to grow for 7 days. And then at day 7, I stimulated them with subthreshold iTBS for 6 hours. And so I was able to do this using these small rodent-specific coils that were developed here in our lab by my supervisor, Dr. Alex Tang. And they're basically the perfect size to just sit directly under the culture plates, so right underneath all of the cells. And so we had two groups or 2 time points that we wanted to analyze. The first time point was immediately following stimulation. So once the 6 hours were up, these cells were fixed immediately to assess acute AIS changes following stimulation. And then our second time point was actually 24 hours post-stimulation. And so with this group, we would then return them to the incubator for 24 hours after the stimulation was finished, before we would fix them. And that essentially allowed us to assess either any long-lasting or potentially any delayed effects of our stimulation. And then after fixing the cells, we ran immunofluorescence microscopy and assessed changes in AIS length and AIS distance from the soma using a custom MATLAB script that was developed at King's College London by the Grubb Lab, who ran some of the original AIS plasticity studies. And so I'll take you through the results now. So here we're looking at the acute changes in AIS structure occurring after our 6 hours of subthreshold iTBS. And as you can see, we found no significant difference in AIS length, but we did see a significant difference in AIS distance from the soma. So the AIS has shifted distally by 18% in our iTBS stimulated neurons versus our non-stimulated controls. And that kind of structural modification, that distal shift in position, is what we would expect to see following chronic increases in neuronal activity. And then looking at the data collected 24 hours post-stimulation, this is actually quite interesting because we actually now do see a significant change in AIS length. And so the AIS is 19% shorter in those neurons that were stimulated with the iTBS relative to our non-stimulated control groups. And again, a decrease in AIS length is the kind of structural modification that we would expect to see following chronic increases in neuronal activity. We also no longer see any change in AIS distance from the soma at this time point. And so that just tells us that those acute shifts in AIS position have returned to baseline by 24 hours post-stimulation. And so those are our major findings. So we are seeing both acute changes in AIS distance from the soma, as well as delayed decreases in AIS length that are occurring at least up to a day post-stimulation with subthreshold iTBS. And so what this tells us is that chronic iTBS delivery, at least at subthreshold intensities, is not only targeting the synapse, but also targeting the AIS, and that these effects are continuing to occur, long past the point of stimulation. So we're still seeing changes in AIS structure in the hours or the day post-stimulation. And so that's something that might need to be taken into consideration for rTMS's application in both a clinical and an experimental setting. But obviously this is 6 hours of iTBS, which I know is a lot more than the standard rTMS parameters currently used in humans, but we are looking at inducing a different form of plasticity here. So AS plasticity is very different to synaptic plasticity and other forms of axonal plasticity that occur on much shorter timescales of seconds to minutes. And so for this reason, it is likely that longer stimulation durations, like the 6 hours used in this experiment, might be what is needed to drive this type of plasticity. And in saying that, it's actually quite exciting, right? Because it opens up the opportunity to explore the use of rTMS in driving axonal plasticity, which could lead to exploring its potential as a treatment for neurological conditions that present with axonal or AIS dysfunction, in which there are currently very few, if any, translatable tools available to do that. And so, yeah, that's it for me. I'd just like to quickly acknowledge a few important people. My supervisors, Dr. Alex Tang and Associate Professor Jennifer Roger, as well as Dr. Jamie Barros, Dr. Darren Clark, and Dr. Eugenia Poe, who have all been really instrumental in getting this project up and running.
Thanks, Emily. You did have a question, but it was, I let you go over a bit because you kind of answered it there about the stimulation length is a lot longer than typically applied. Just very quickly, in a one word maybe, do you think you'd see anything with shorter stimulation lengths?
Potentially. Yeah, potentially that's what we're looking at now, but okay. I don't think it will be as short. Yeah, no worries.
Great. Thanks, Emily.